Thanks, sir.
Today we have a very senior professor, Dr. Jyoti Yadav from the Biotechnology Department. She is the head of department of Shivaji University, Kolhapur. She has done a lot of work on different areas of biotechnology, including bioremediation. So today's talk will be on bioremediation, and we'll really enjoy the talk. Now I request Sumit to give the introduction.
Yes, thank you, sir. Sir, am I audible?
Yes, you are.
Okay. Good afternoon to all, and especially good afternoon to Professor Jyoti. I am very glad to give introduction of Professor Jyoti Yadav, Senior Professor, Department of Biochemistry at Department of Biotechnology, and she has nurtured his department into a center of academic research excellence and has received more than 15 crore funding and published 200 international publications, 20 books chapters, around 8,000 citations, 51 H-index and 121 I-10 index, and 2 patents and guided 30 PhD students. And she is the world's leading scientist in the area of Parkinson's disease, Alzheimer's disease, and phytoremediation. She has made several noteworthy contributions on bioremediation of textile industry, effluent using microbial plant and transgenic hairy roots. And this approach has been employed to treat effluents from industrial waste. This work has been highly appreciated by Department of Biotechnology, New Delhi. Professor Yadav was program investigator of interdisciplinary program for life sciences, which was successfully completed. And now she is in the top 2% world scientist in biotechnology and environmental sciences. Report released by Stanford University, USA, consecutively for 3 years. Scientific communities like Scopus placed her at top 4 for research publication in the area of decolorization. According to World Scientists and University Engineering and Technology Engineering Ranking 2021, she is 7th ranked scientist at Shivaji University. She has been awarded with the Women Scientist Award constituted by Biotech Research Society of India in 2012. She is a member of National Academy of Science India and fellow of Maharashtra Academy of Sciences. Recently, he has been awarded with Best Teacher Award by Shivaji University, Kolhapur, also bestowed with Brand Kolhapur Award. Professor Yadav has delivered 100 more invited talks in many national and international conferences, and she is a reviewer of 66 international journals and member of around 80 national and international advisory committees across the country. So I hope this lecture is very knowledgeable for all participants. Thanks.
Yeah, thank you so much, Dr. Sumit. Am I audible?
Yes.
Yes, yes.
Yes, you're audible. Professor Yadav, I think it is hanging. It is hung.
Sir, she started his screen.
Hello?
Yes, you can do one thing. You're okay now?
Yes, sir. Am I audible?
Yes, you are.
Yeah. Because in between, I am not listening to anything from your side. Shall I start?
Yes,
Okay.
You start.
Yeah. Okay. Thanks, Dr. Sumit, for a very nice introduction. First, good afternoon to everyone. It's my privilege and honor to be here on the prestigious dais of MBSI, and I'm very thankful that at the outset I would like to thank the organizers, especially Senior Professor A.M. Deshmukh Sir, President of MBSI. Uh, Actually, he has created a very nice group, and after that I came to know that lot many activities are going on, and immediately I became a part of this very good prestigious society. I'm very happy, sir, that you have taken a lot of aspirations for arranging such a good platform for my work. Especially would like to thank Professor Swarmjit Singh, President of MBSI Chandigarh. He is ex-director of SAS Polyclinic and ex-head of Environmental Biotechnology CSIR, MTCC Gene Bank. Sir, thank you very much. Very nice to talk to you all. Yes, I am Professor Jyoti Jadhav, head Department of Biotechnology, and my area of research is bioremediation of textile dyes and effluent. Since 2004, I'm working in the particular area of bioremediation. In the beginning, actually, we came to know that lot many studies are required in the area of textile dye effluent treatment and all these things. Many researchers, they are working on the decolorization process, but exactly the mechanism of work and the enzymatic process what involved in has not been elucidated, and that's why we have decided to work on this bioremediation area, and we started at that time with the yeast as a first microbe for that. And today I'm very happy that I'm giving the lecture. I have chosen this particular topic on the MBSI platform. In short, I would like to discuss here that what bioremediation is. Actually, bioremediation means the technique which employs the living thing like bacteria, fungi, and various plants also for the removal of pollutants, textile effluents, and various toxicants from the environment. And basically, microbes are playing a very important role, and I would be emphasizing this the role of microbiology in the cleanup of these environmental parameters. And as you know that I'm especially emphasizing on the dyes and pigments because in India, uh, different lot of many textile industries are doing work for the dyeing and all these things, and more than 10,000 different pigments and dyes we are using annually. If you see the statistics for the annual production, then statistics is very high, and the textile industry effluent is discharging around 2 lakhs of 2 lakh tons per year of this textile dyes in the effluent. So obviously it will be hazardous to the humans as well as the living organisms in the soil and water. So we should think in that way, that whether we can do something as a scientist, and we started working into this area. And here are some statistical data that what exactly the dyes and pigments, what is the economics of dyes and pigments. And because why this happens, because we all love to live the colorful life, and the colors are most important part of our life, and that's why everywhere we need the color, and in the form of dyes, in the form of many even edible colors, etc. And actually this is my working area. So why it is necessary to be the bioremediation? Yes, the bioremediation, everywhere we are talking about the chemical processes are there, physical processes are there, and I'm not going into the details of the physical or chemical process, but I know that these methodologies have certain drawbacks, and the bioremediation is obviously the biological tools we are using, and it is a safer than other method, and actually it is a cost effective also. And whatever contaminants which we are degrading, basically toxic compounds that are completely they are degraded into very simple molecules, and which can be used as a carbon source for many of the plant living organisms. So that is the need, that why we chose the green approach. So this is the current scenario that everyone is knowing about, that whenever we are passing across with any textile industry, we are seeing the lot of dyes, number of dyes, colorful waters are been thrown into the rivers, and many they may cause several problems. So this current scenario actually facilitate us or inculcate us that whether we can do something in this particular area. So I would like to emphasize here very some brush up that how many different types of dyes are there in the atmosphere. Is my PPT is seen to everyone, na? Hello.
Yes, yes, we can see everything.
Yeah,
Yes.
please. Yeah. So these are the different types of dyes that some of the dyes are natural dyes, then some belongs to plant pigments, some are synthetic, acidic dyes,
Hello,
basic
madam.
dyes.
We
Yeah.
cannot see your PPT. We can see you only.
Okay, okay. I'll share again, sir.
You
No
share it
problem.
again.
Yeah. करते the share. तसं का होतं मध्येच? Sir?
Yes, now okay.
Yeah, earlier slide. Have you seen this thing?
Yeah, I can see the slide, but it is small. Can you make it big?
Yeah, big. Actually, my screen is showing big, sir. Okay, I'll again try.
Okay, okay, please.
Full screen, full screen. तुम करना. Full screen. Full screen करना. Now?
I cannot see. I can see you only.
Oh,
You can
oh.
share the slide.
असं होत नाही ना, तसंच करावं लागणार.
बरं, ठीक आहे, करते. Entire screen कर.
Yes, make it big. Yes,
Yes, it's
now
okay.
okay.
Okay,
Now
sir. Yeah, yeah. So this is about the current scenario, and yes, we can see that there are number of dyes which are available in the market. Some of natural dyes and some are the acidic, basic, and disperse reactive. And obviously, if the dyes are of different characteristic property, we need a different type of approach to remove these dyes from the environment. So that is the way that yes, the physical methods, some scientists are using the physical methods, chemical methods, and biological methods. And biological methods, we tried actually bacteria, fungi, algae, plants, and their consortial system. So what is bioremediation? As I thought the number of students are also as a group of MBSI. So I thought that what exactly the bioremediation means. It's actually the mechanism which involves the various facts that the phyto extraction, phytostabilization, phytodegradation, rhizodegradation, and phytovolatilization. Actually, plants are mainly responsible for all these types of remediation process along with the bacteria, and that's why it is a very big task what we observe, because initially we are working only on the bacteria and some varieties of bacteria. We have isolated different varieties of bacteria from the textile effluent site, then fungi, many other microorganisms, and thereafter we also tried certain plants, and we came to know that yes, plants are also having the same capacity. And after that we came to know that the plants have capacity because of the rhizospheric bacteria. So this is a very interesting fact we observed long back, and yes, and that's why we have used the different plant and bacterial consortium for the degradation of various different textile dyes from the effluent. So these are the applications of bioremediation, that many toxicants, that like heavy metals, then chlorinated solvents, then pesticides and textile dyes, many more things we can remove with the help of bioremediation. In our studies, we use for the bioremediation of textile dyes, we generally prefer the plants, basically the role of plants with the bacteria, fungi, as well as yeast. So what type of, you know, that I have already told you that our lab is a very famous lab in the area of bioremediation. I'm working with two senior scientists in our department, and around 35 more or 40 more students have got their PhDs in the area of bioremediation, and they are working in a very good place across the globe. So obviously, yes, our entire lab is working on the bioremediation, and we have used the microbial species as well as plant species. And here is the list of certain bacterial species and plant species what we have used for the degradation of textile dye. And you can see the number of publications from our area, and you can check it out, what is the potential, what dyes they are degrading, etc. So this list gives you the clear-cut idea that yes, n number of bacteria we have used in our laboratory in the primary studies in order to transfer the technology to the lab. So initially, I said that our main focus is the bacteria, but somehow we felt that the bacteria we cannot use at the actually site. So we think that can we use the other methods also for the degradation, for seeing the for studying the dye decolorization. So initially we don't know about what type of plant can be used and all these things. So we started with the various plants, flowering plants we generally use, and amongst this I can emphasize here is the Tagetes petula is the most important variety we have observed that it is having a good potential, and it is also withstand for very different types. Typha is also then, and then we started working on the wild plants also. So I would like to emphasize here that what type of flow chart we are using and what are the standard protocols what we are using, because most of the researcher, I thought in the area of microbiology, they might be referring my papers, but I want to give you that what flow chart we are following in our laboratory. That initially we are screening the different microorganisms or plant from that particular site, for example, cottage industries or the major big industries which are dyeing industry, textile industries, and this may be especially while selecting this plant, we are considering this tree may be non-edible, having the massive root system, and it must be fast growing. After that, in order to check the potentiality of this plant, we have checked this plant. We have used the pure plants in the test tube in the test tubes also. So in order to see the exactly what plants are doing, whether the plants are capable of doing the bioremediation or they require any bacterial help, this also we have checked that whether plants are able to do or not, and for that purpose we have used the tissue cultured plants also. And when we came to know that what type of enzyme systems are required, and specifically as for the any type of xenobiotic compound, mostly oxidoreductases plays a very important role. And here I am emphasizing that the enzymes which are responsible for the degradation of textile dyes are lacase, lignin peroxidase, azoreductase, tyrosinase, NADH DCIP reductase, as well as volatyl alcohol oxidase. So in our laboratory, if suppose any of my research colleague or research student is working on the particular dye or particular microorganism, first we check that whether the plant or bacterial system has this particular enzyme activities, because all these enzyme activities, their assays were standardized in my laboratory, and first we are checking the activities of these enzymes in the particular species. After these activities, we also perform the several analytical techniques we are using for the confirmation of degradation of very complex structure of dye, and whether they were converted into the toxic compounds or the plain or the complete mineralization. So this is the main aim, that as I'm saying that this is a bioremediation, that means it's an enzymatic degradation, and this is a very specific degradation. And obviously we are getting a very positive results, that we are getting the final products which are very non-toxic, and that's why it's a very effective technology nowadays. A number of industries, they are using this phytoremediation and bioremediation technology. And obviously we also perform the final product after performing the different analysis like FTIR, GCMH, HPLC, HPTLC, etc. We also predict the dye degradation pathway, that the complex dye, whether it is converted into simpler form or whether it contains any other toxic material. And if I am saying that yes, we are seeing the non-toxic material is produced due to the biodegradation of any textile dye, we need to check the toxicity. And in our laboratory, we did the number of toxicity analyses like phytotoxicity, MTT assay, toxicity on various aquatic animals, And also the molecular techniques we are using, like the comet assay and so on, to check the chromosomal aberration changes because of this dye solution, means the dye before degradation and after degradation. And obviously the work we are doing is not for the laboratory work. Obviously it is to be necessary that whatever work we are doing, whether it will be helpful for the society. And in our laboratory, number of plant species and number of microbial species we have studied. And finally we constructed the laboratory scale bioreactors. And after successful implementation of the technology at the laboratory scale, we also use this technology in the for the construction of wetland. So this is somehow flow chart. If anybody wants to do work in this particular area, you can check it out, what publications we have and what type of flow chart we are following in our department. So first, one by one, I'll give the sum up of my work, that how many different types of plant remediators we have used. These are the few names of plants, the names of dyes, how much percentage decolorization and what time they are degradating in. And also we found that these plants are also very good bioremediators. They also individually remediate different types of dye in a normal way. So these are again few glimpses that how these decolorization experiments were carried out in my lab. You can clearly figure it out that before addition of plant, how what is the color of this particular plant, and after addition of, after going this plant into the these things, how the color is immediately changing. These are the few examples here. So yes, obviously when we are saying that the decolorization occurs, means what are the biological means behind this decolorization. And here this particular table is giving that number of plants showing the activities of these particular enzymes. Then if this particular table is showing that there are few plants which are showing there are the plants which are showing very few activities, not necessary that all the activities should be present in one single plant. That means, for example, if we have taken the example of Tagetes spicula, then it is showing only the lignin peroxidase activity, azoreductase activity and DCAP reductase activities. Likewise, Bromia malconi, it is also showing the lignin peroxidase activity, then tyrosinase activity and DCAP reductase activity. That means this particular table is showing that the plants are very specific towards the dye. Not all the plants are showing all the activities, and that's why we have strategically developed that whether which type of plant is useful for what type of dyes. It decides that what type of enzyme pattern they are having before degradation and after degradation, and accordingly this particular type of studies are useful for development of strategic planning of the textile effluent treatment. So after the evaluation of how you can see the evaluate that whether the dyes are degraded or not. So definitely I'll take the examples that we are we set the certain experiments where we are seeing that the before decolorization, what are the colors and how many days they are requiring in. Here I am giving the examples of one plant list F dicomata, where we have made this consortium of this with the Saccharomyces cerevisiae, and we observe that when we individually treat the dye Ruben GLF, then individual degradation capacity of plant is also lowest, and the Saccharomyces cerevisiae is also lowest. But when we combine these two together, obviously they are giving a very good results, and the percentage degradation of the dye is also high, and the number of days required for the degradation processes are also high, very low. And the third point here, I have specially given the title that the combinatorial therapy, because earlier what we are seeing is that earlier we are using only plants, and when we grow this plant in an open atmosphere, obviously the microbial growth is there, and there is a synergism between the plant and microbe, and they are also giving the some fast decolorization process. But after some time we found that after some time, if we are continuously feeding some dyes, toxic dye solution to the plants are not surviving very properly because they are continuously coming in contact with the very toxic molecules like dyes. So in order to solve this problem also, we thought in the third way that whether we can able to provide some feed to the plant, and along with that whether we can encourage the bacterial rhizospheric bacterial growth, and for that purpose this triple consortia concept came into our mind. And here I am showing few results of them. So generally what happens when we study any treatment of any effluent, or when we then the ultimately the parameters which are came into picture are COD, BOD, ADMI, pH, turbidity, TSS, TDS, and also the bacterial growth. Because most of the physical scientists and chemical scientists, they are doing the dye degradation process, and they are simply seeing that if the color change means the degradation of dye. But according to me, only the color change, it doesn't matter. Sometimes these chromophoric groups which are present on the dye, even the small pH change in pH, this color is become colorless. It doesn't mean that this actually degradation takes place, and that's why the biological concepts come into picture. And when we are seeing that whether this concept is degrading by certain things, then actually how much the COD is changing before treatment and after treatment. So these are the parameters like COD, BOD, ADMI. Continuously we are checking in our laboratory, and if this particular table we can see, then the values of COD, BOD before treatment, after treatment, individual treatment with the plant, individually treatment with the Saccharomyces cerevisiae, and treated with the consortium of the FD and SC. Ultimately it is giving the values which are normal one, that we can easily use this water which is treated with the help of this consortium for the can be directly spill it out in the water bodies, or we can use this water with the help for the agriculture purpose. So these values are very, very important. And when we are studying, we are not just stopping that what type of enzyme activities are there, and or how much percentage of degradation occurs there, but we are looking for the parameters which are actually required for the MPCV or CPCV values, which are recommended values that the water can have, and this water can be spill it out in the water bodies. So this is the special I can say the positive part of our study, that we are taking this care of these parameters also. Along with that, we also check the enzyme activities, because as I earlier in a flow chart I said that whenever we are using any bacteria, any fungi, or any plant, first we need to check the what type of activities they are having. And merely we are observing that some bacteria showing some enzyme activities, some bacteria, some plant showing some other enzymatic activities, and all the activities were not present in single bacteria or single yeast or single plant. So that's why we need to combine together in order to have all the bacterial pool. They are having these enzymes which are directly taking care of all the toxic materials which are present in the effluent. Because in effluent we are having the number of combinations of dyes, like 20 dyes or 24 dyes, or many other things are also there, and we need to take care of that toxic materials or toxic compounds with the help of the different system, when they are working together in the form of consortium, like a plant bacteria consortium or a plant yeast consortium, etc. Then here this particular table is showing that when we see in the enzyme activities in FD and Saccharomyces cerevisiae, then most of the activities are not present in FD, most of the activities are not present in the Saccharomyces cerevisiae. But if we combine together these activities, basically they compensate with each other, and then finally they degrade whatever dye stuff which is present in the effluent. And this dye stuff is obviously it is converted into some neutral molecules, or the primary amines or secondary amines. They are decreasing their BOD, COD, etc. values, and ultimately they are doing the work of bioremediation, converting the toxic molecule into the simpler form that can be easily assimilated or can be used directly as a carbon source by different bacteria or different plants. So how one can detect, how we are detecting these things directly by going through the several spectra, FTIR spectra. This particular, I will not go into the details, that you can easily read it out, that yes, what these spectras are. But generally we do the spectra before spectra of dye, as well as the spectra of the degraded metabolites. This is also the HPLC chromatograph of the control dye, and the dye which is treated with the FD, dye which is treated with the Saccharomyces cerevisiae, and the dye which is treated with the consortium of both FD and SC. And these particular analysis, the analytical techniques clearly indicates that the efficacy of the individual entity as well as the efficacy of the consortia for particular dye. So that type of scenario everywhere we are following, to know that what strategic mechanism or what is the mechanism followed by the individual plant or bacterial species or yeast species, and when they are in the consortium, what would be the fate of this particular molecule. And we also do the we also predict the pathway, because when we came to know that yes, degradation occurs, then enzymatic activities are increasing, then BOD COD levels are going to decrease, ADMI values are going to decrease, and we are also seeing that there are certain spectra showing that these peaks are different peaks from the original world. Then it is but obvious that what is the metabolic fate of this particular dye. And this is the way that with the help of induction pattern of different enzymes like veratyl alcohol oxidase, lacase, and etc., we also predict the metabolic pathway. And this is the reason that we are getting a very good publications in the very high impact factor journal having the impact factor 10, 15, 18, etc. So this is possible that as we deeply study the enzymatic pattern, the enzymatic pattern will give you the clear-cut idea that how this molecule is being degraded and what is the final product. So obviously once we did this metabolic fate and all these things, the question arises, the whatever metabolites which are produced after the degradation and after the checking, after consortia, whether they are toxic or not. And any scientist, yes, we cannot say by seeing into the spectra that this molecule is toxic or not. So in our laboratory, we also standardize the different toxicity analysis. Especially we initial level, we did the phytotoxicity when our labs were not that much developed. So initially we are doing only the phytotoxicity. Then thereafter we also standardized the MTT assay. We also did the genotoxicity, and we also nowadays we also did the fish toxicity, bivalve toxicity, etc., etc. So one by one, I'll tell you that when we are saying that the dye is toxic and the product is non-toxic, we need to declare that, and we need to code these values with the help of the plant growth. If suppose any molecule is toxic to the plant, then obviously it hinders the plant growth, and if it is non-toxic, obviously it increases the greenery and the appearance, physical appearance of the plant. So initially while doing the plant toxicity, we also did the plant pigment analysis like chlorophyll A, B, and here are the few pictures which are showing that the chlorophyll content of the plants when we apply the treated sample is more, or if it is very less, it is decreasing when we give the sample with the dye to the cell. So these are the there are number of tables, and in the number of publications from our lab, you can very easily see it out that what type of these things we are doing. So in a phytotoxicity analysis, initially we are very strict towards the certain plant species like Triticum aestivum or Phaseolus mongo, and we are seeing at that time only the germination rate and their root and shoot length. But now onwards, we are doing all the plant physiological analysis for getting whether the toxic product is toxic or not, whether it is whether this water is whether this water can we spill it into the water bodies, or can we use the same water to the farming. So these types of analysis of the control dye, treated textile effluents, or untreated textile effluents routinely carried out in my lab. And here are few pictures clear cut showing that the plants which are simply growing in the distilled water and in the dye, even they won't be able to sustain, because these are the control plants, and we have added the all seeds are same in every pot. But here it is showing clear cut that when these plants are treated used with yeast, then the number of plant generation rate is more, then the survival rate is more, they are looking very good, and others are somehow defective. These are the few picture clear cut, they are showing that if when we are using only distilled water as a for plant growth, then this is the picture showing that how they are growing. When we grow the same plants, the plants in the dye, they are also having the stunted growth. But if we are putting this with the consortia with the help of only dyes and this, then they are showing a very good root length as well as the shoot length. So this obviously clear cut indicates that the degradation process what we are following are user friendly, they are biodegradable, they are environmental friendly, and can be used on a large scale. Not only that, we also studied the anatomical studies of root cells, because actually what happens when we study the actually dye, what happens when dye it is enter into the root cells. Initially this dye entering on the outer surface, and after some time this dye is enter actually inside the cell. And as I said that these plants are also having this potential to have the some enzymes are get induced after when the root cells are exposed to the dye. Then after few hours, like 72 hours or 48 hours, the enzyme induction occurs inside the cells of root as well as stem, and they will take care of the dye which is penetrated inside. And finally after some time we found it that the dye become completely vanished from the root cell. So not only we are seeing that the effects of our dye effect of dye on the plant externally, but internally also we studied this inside the root cell. These are the few pictures showing that how the dye is entering inside, and particularly this dye is entering inside the root cell, how the enzyme induction occurs and how this dye is degraded inside. So this picture clear cut indicates that yes, this is a biotransformation. This is not simply a pH change or something else. And not only that, these dyes are entering inside, they are degrading inside into non-toxic in the presence of dye, and this is a very good example purpose. Secondly, we also studying the cytotoxicity test, because if we are using, they are having the carcinogenic effect, or they are having a very bad effect on the plant cell regeneration. But when
Hello madam.
Yeah.
Again the PPT has gone.
Yeah, gone again. Okay.
Gone again.
Okay.
Why this goes away suddenly?
I don't know, sir. This time this happens only first time to me.
I think some internet problem.
Yeah. In our area, because of this rainy season.
Yes, I think so.
Yeah. Connectivity problem.
Yeah, now coming. Yes, make it big.
Yes.
Okay.
Earlier, have you seen this?
Yes.
Yeah. So this particular diagram shows you that how the physiology of the plant get changed when dye enters inside. We also tested with the cytotoxicity. Then there are a number of plants we have used. You can check it out with the number of publications. Then animal toxicity, as you know that most of the water after treatment it is spill into the water bodies. So obviously question comes that whether it is going to affect on the animals which are present in the water bodies like fishes and many more. So definitely it is going to affect, and you can clearly visualize that how this transparent fishes are converted into colorful fishes. So how it is going to be affecting on the creatures? So if suppose this is the control when the fishes in the lab, when we grow into the controlled water, and in the dye solution of minimum concentration only, it is not that much high concentration, but even though in the minimum concentration they are dying, or they are affecting the metabolism and entire physical appearance of this particular fish species. And after treatment also, when we treat the same dye, and we float we keep these fishes into this particular things, then they are very normal. So that type of animal toxicity we also carried inside our lab, and we also done the liver histopathology of this exposed fish, many more other organ histopathology we also doing in our laboratory. These are the few pictures clear cut showing that how the toxicity analysis is been done, and how their internal gill structure can be studied, how it is affecting when the controls, and it is to be compared with the control as well as the treated dye. When the fishes are exposed with the controlled water and treated water, they both are same. But if we expose the same fishes to the dye, then their internal gill structure is also get disturbed. So this is the clear cut indication that how dyes are toxic to the animals. Here also the same, I will not going into the details that how these control things are matching with the treated effluent. Means in the dye exposed bivalve, the the internal structure is disturbed, and while in case of treated effluent, it is as good as control.
Hello, madam.
The specific dye, whether it is toxic or non-toxic before and after treatment.
Madam, again your PPT has gone.
Oh my God. Okay, sir. Again, I'll.
I think there is some problem with the internet.
Yes, sir. Is in this area because of some thing, na?
Rain.
Our net is going, and I am on land, na?
Right.
Yeah. Sorry.
No problem.
Okay. So,
Yes.
considering these
Make it big. Hello, make it big.
Yes, actually it is big. Yes, I'll try again. Is this okay, sir?
Yes, not yet big.
Again, I'll share. No problem.
Now gone, completely gone.
Yes, I'm sharing, sir. Done?
Not yet big. It is small.
Yeah, last 2 days back also, one meeting, same thing happened, sir. In probably there might be a certain deformities in the connectivity of the university actually.
I see.
Yeah,
You can
it cannot be possible.
You can continue with the small one, no problem.
No problem, na, sir. Okay, shall I try once again?
Okay.
One more time. Yeah.
Excuse me, ma'am. You just press F5 for full screen.
Yeah, I'll try. F5.
Now again the PPT gone.
Yeah. I'm trying, sir. Please wait. Share करो. हाँ कर. Okay, sir.
Yes. Okay.
Full screen.
Yes. Okay.
Okay.
Yes.
So with this research background, we decided to go for the bioreactor designs, because in order to see the sustainability of the lab experiment, necessary to check the efficacy of the microbial species or plant species. We have designed a number of bioreactors in our laboratory, and these are the few designs and how they are going to work in our laboratory as an individual plant or in the form of consortium. And initially we designed the bioreactors with the help of flowering plants, and thereafter we also designed the same plants with the consortium and the plants which are directly aquatic plants, which are sustainable actually. So these are the few plants where the soil layer is in between, and we are continuously adding the different types of dyes or textile into that. And ultimately we are checking the continuously the parameters which are required from the running out water. So this is also the second live this bioreactor. Actually you can see that how it is to be looking up, the roots are there, whether this particularly which are immersed into the inlet, and they are giving a good efficacy. And these bioreactors are running in the lab for years and years, means 1 2 3 4 years, while the one single student is working in each individual plant, and they are studying the detail of these things. So the strategic theme behind that is that whether the plants which are sustained in the laboratory, whether they would be able to sustain in the field or not. So this big bioreactor we have prepared, and actually the name, the work of our lab is been displayed over in the DBT site also, because the data we have presented in some meeting over this the environmental biotechnology meeting, and they are very this work is appreciated, and they have put this bioreactor designing and the work which is done in our lab on the DBT site on the environmental biotechnology especially. So here I want to show you that these are the certain parameters which we are looking for before and after treatment, and you can see that how significantly the required parameters are decreasing after this particular effluent is moving through this bioreactor. Here this particular bioreactor we have made with this coupled reactor with this aquatica and hydrofolia, two different types of plants we have used here. As I said that not only these bioreactors, number of bioreactors were constructed, and every PhD student is working only on the bioreactor. But somehow after some time we came to know that these plants will not be able to sustain only if we are continuously supplying them with the effluent. Yes, they are sustaining, but this is not good for the plants also. So how to sustain all these things? So the third concept came into my mind, and last 3-4 years I am working on this particular, that how you can use, how you can enhance the capability of plant. And obviously for the enhancement of the capability of plant, microbe like yeast did a significant role, and I am happy to share here my results over here is that how this yeast is being used here in the triple consortium, and how it is to be beneficial both for the plant as well as increasing the efficacy of the textile dye effluent treatment. So here is the one bioreactor designed by one of my research student Rahul Patil. Rahul Jadhav, he is working on this triple consortium. He has used yeast as a consortial material, which is providing a best base for the growth of bacteria of a specific type, as well as it also fed the plant. And this publication, our entire publication, recently we published this paper in the environmental pollution research with a 10.9 something impact factor, and this is very good promising result we observe here. So here are some bioreactor he has made with, and we have checked the efficacy since last 4-5 months. We are checking the efficacy of the this bioreactor, that how these plants are growing very nicely and happily in the atmosphere when we supply the few quantity of very few grams of yeast when we are supplying. So what exactly happens inside? These enhances the bacterial growth, rhizospheric bacterial growth. It is also feeding to the bacteria, it is also feeding to the plants, and both these then happily work together for the effluent treatment. And here are the sub few example that immediately we are getting the decolorization within few days or few hours. And these are the pictures showing before and after treatment method. Here is the one simple design. We are also increasing the sophisticationness of this experimental design thereafter. And this is clearly showing that the yeast and bacteria sorry, plants. Yes. Then next thing approach, then as I said that we have already started, we have already shifted our technology for the construction of wetland in our area. And this is mostly our approach. And here there also we did the constructed wetland in around 10 acres of area. And this is also nowadays because of this COVID and all these things, our project is also over. But this constructed wetland is working till today. Here are the few glimpses that how it is needed that we need to take care of this textile effluent which is being flowed into the certain areas. Then this is the current scenario here in the HRTS Kagal, because we have made some MOU with them. And we are very fortunate to have to them that they have given us the space of 10 acres of area where the plants which are studied in the laboratory, we shifted all these plants to the this particular area. And these are the few pictures showing that the before and after treatment, how the effluent is coming and how it is to be detoxified. But before directly transferring transfer of this plant, it is not possible directly transfer the plant from lab to land. We have constructed certain lagoons, and these are the few pictures that certain types of plants which are being studied in the laboratory. We have first acclimatized this plant into the specific lagoons having the 60,000 liter of capacity. And here this particular the table will give you that what are the parameters initially present in the untreated effluent, and what are the parameters after 96 hours, and what are the parameters after 192 hours. And here I deliberately added the MPCV standards, what they need to throw the effluent or somewhere into the water bodies, or to provide the same water to the this particular for the farmers. So definitely these norms, we have reduced the toxic norms to the neutral ones as per the MPCV standards. So I think this is a great achievement of my lab, that we can be able to achieve that much standard by using the different consortia. So here are one consortia with Ipomia aquatica, lagoon, then second lagoon of Salvinia molesta, then third lagoon of Typha augustifolia. And these all lagoons also were constructed in MRDC Kagal in our HRTS area. As sir is saying that he has gone to the Canary Mud, and this Canary Mud is the other side of road, and this HRTS Kagal, our construction side, is this side. So these are the some side pictures here. These are the few pictures I'm showing here, is that the lagoons. We also because in this particular area there are two lakes, small lakes are present. So here we also made some floating phytobeds, and we would be able to decolorize the lakes which are filled with colored water. This is also a great achievement, and it is sustained. This has been sustained 3-4 years at that time. So this can be a good sign that this technology can be developed and can be used everywhere for the textile effluent treatment process. These are the few pictures again. Phytobed picture. And obviously every scientist is doing work for both reasons, for getting publications and for getting the technology to be useful for the society. And I can lastly say that yes, our group along with my seniors and my juniors, we all are successful in both ways. We would be able to publish a very good publications in a high impact factor. You can see all these are very good journals and number of publications. And Here is my research group. Especially, I would like to thank Professor S.P. Govindwar and myself. We have started working in this particular area from 2004-5, I think. This is our first paper in yeast that we published, that yeast can be used as a tool for a bioremediator. And thereafter, we have published up till now 285, more than 285 publications and number of book chapters. Recently, we published the book chapter only on the bioremediation and contributed by most of my research students. So see the potential of this particular work, how it is to be. I would like to thank Professor V.A. Bapad, because initially myself and Professor S.P. Govindwar, we are using only microbes or bacteria and fungi. But there are certain limitations with these microbes. But after entry of the plant biotechnologist, Professor V.A. Bapad, he has inculcated.
Excuse me, ma'am, you are not audible. Some technical problem is there.
I think he has finished most of the talk, so we can conclude the talk now. 1 minute, I will just talk. Hello, Madam Jyoti. Hello, Dr. Jyoti. I think we can conclude and again call her some other time. Hello, Dr. Jyoti. Hello, Dr. Jyoti.
Sir, I think completely disconnected audio.
Okay, it was an excellent talk by Professor Jyoti. The PPTs were very vivid. We understood every para what she spoke. It was an excellent talk by Dr. Jyoti. Now I request Dr. Sumit to give the vote of thanks.
Yes, good afternoon to all. Good afternoon to all. Some technical error. Dr. Jyoti Yadav left our this webinar. So, Dr. Jyoti expressed their thoughts on bioremediation and combinatorial technique as green approach for the treatment of textile dyes and effluent. And their teamwork and her work is very excellent regarding society, and their work is very helpful to create some specific bioreactors with the help of microbes. So thank you, Dr. Jyoti, and thank you to all participants. Thanks, sir.
Thank you very much.